we speculate that it might help in activating the B cell. I'm not going to put that on the test though because it's just speculation. To draw someone's blood and look at their antibody count, the most abundant antibody in a person's blood would be of the IgG class. In fact, it make up about 80% of all the antibodies in someone's blood. One nice thing that's really cool about IgG is that it's small, and so it can get into small places. It diffuses well. In fact, it can even diffuse across the placenta and plays an important role in protecting the fetus. So antibodies usually last about 6 months. So after a baby is born, the baby is protected by the mother's IgG antibodies for about 6 months. Does that make sense? Because the mom produces IgG antibodies and it will cross the placenta and get into the baby's blood. So when mom is exposed to something, she makes antibodies, and those antibodies are there in the baby. Now, the baby's not producing its own antibodies, right? The baby's not producing its own plasma cells. And so this type of immunity, where you actually get antibodies from somebody else, is called passive immunity. Okay? Passive immunity is when you're not exposed to the disease, you're not exposed to the antigen yourself, somebody else is, and they give you antibodies. When you are exposed to the antigen, or you're exposed to the disease, or you're exposed to a vaccine, and that causes you to make the plasma cells, then that's called active immunity. We're going to talk a lot about this in the next chapter of immunization, but make sure you know the difference between passive and active immunity. So do you want me to say that again? So passive immunity is when you get antibodies from somebody else who's made them. You've never been exposed to the antigen or the disease yourself.
What's another example besides
Yeah, so sometimes what happens, if you're going to a country or a place that you have never been exposed to, so like if somebody was, if someone was coming here to the United States and they hadn't been exposed to a lot of the diseases there, one thing that you can do is you can get a serum that has a bunch of antibodies from a whole bunch of different people. So let's say that what I could do is I could get blood from all of you guys and concentrate the antibodies down into a single shot, and then I could give that to somebody else that's coming here that's never seen all the diseases of Rexburg. And that would give them a lot of protection because you guys have already built up antibodies to the diseases here. Missionaries often get these shots. They're called gamma globulin shots. Their nickname is peanut butter shots because they're so thick that when you put them in, it hurts. But what it is just a whole bunch of antibodies that are taken from the people, the demographic of that area to give you, and again, those shots work for about 6 months.
So passive immunity is basically receiving antibodies from someone else, whereas active is you receiving antigens creating your own antibodies.
Yes. Active is when you see the disease yourself or you produce the plasma cells yourself. Yeah.
Did you say active is more effective? Or more
Active is definitely more effective as long as you do it before you die. Right? I mean, so that's why you would give somebody the passive if you're worried about a really disease with high mortality rate that will get them down before they have an immune system.
What's that AD there of all the workers is transfer AD across?
Cross plus have an important role for design person. Is this one?
Yeah, that one.
Oh, AB. Oh, sorry. AB is an abbreviation for antibody.
Oh.
Good to know. abbreviation for antibody.
What was the bone marrow transplant? Like, is this extracted?
Bone marrow transplant, you're actually providing the cells. Yeah, because that's where all your immune cells hide out. Your plasma cells, they'll start out in the lymph node, but they'll go to the bone marrow. That's why you get bone marrow transplant.
Both parts?
Did I skip a slide? Did I tell you, you know what, I think I skipped something earlier. Can I just go back and make sure it has to do with, I don't think I talked about this slide, did I? Okay, so you're going to divide this, we've divided this antibody by its top and bottom by its different functions. Turns out that we've named the top and the bottom. We don't really call it the top and the bottom, because that's too easy. The stem portion, okay, let me just, how they did this, they wanted to know, early on when they discovered antibodies, they wanted to know how it functioned. So one way they did it was like, well, let's just cut it up and see. So they took these enzymes and they started cutting up the antibody. That's how they figured out there's 4 different chains, for example, a small chain. But it turns out if you just slightly digest the antibody, the first thing that's cut is right here. It cuts the stem portion off from the top portion. And when they cut the top portion off, they figured out what the job was of the top portion, because it was still able to bind antigen. So this top portion was called Fab, or the fragment that binds antigen. It's an antigen binding fragment. The ABF didn't really sound good, so they put the F in front. So it should have been ABF for antigen binding fragment, but Fab sounds so much better. They just did that. So anyway, this is the antigen binding fragment called the Fab. And so whenever you see this on the slides talking about the Fab region or something, that's the top region. Now the bottom region, they cut that and they couldn't figure out what the heck it did. Because they didn't know about complement, they were just barely figuring out phagocytosis and stuff like that. But they did know that when they froze it down in their freezer, it made super cool ice crystals. So FC stands for the crystallizing fragment. Like this doesn't help. Okay, so whenever I talk about FC, it has to do with the stem. Whenever I talk about Fab, it has to do with the top one. That's important because on the exam I talked about Fab and FC. That's why I said, oh man, I didn't talk about that. Yeah.
On IgG, is that the one that attacks the baby if the mom is negative, blood is negative, and the baby is positive?
Yeah, that's right. It used to be a big problem than it is now. Now we have pretty good drugs. Right, ready? The next one. Oh, sorry. IgG also activates the complement system, but as we saw, it's not as good as IgM. Okay, you have to get a lot of IgG onto a cell before the antibodies are close enough for C1 to attach between 2 FC portions or stem portions of the antibody. IgG is also a good option in that phagocytes have receptors for this FC portion, or the stem portion. Questions about IgG?
So you just said passive immunity is only for about 6 months usually?
Yeah, because antibodies only last about 6 months. And as time goes on, it'll dwindle down to so it's a lot better at 2 months than it is at 5 months. Okay, the next antibody, some of you will really be interested in this time of year. IgE, as you can see from this picture, if you're having hay fever problems right now, summer, is because you're producing a lot of IgE antibodies. So IgE mediates the immediate hypersensitivity reaction, in other words, allergies. Hay fever, asthma, hives, even anaphylactic shock are all caused by IgE antibodies. You're like, wee, yay, let's produce more IgE antibodies. Right now you're asking, why the heck do we have IgE antibodies? Yeah, as your eyes water and you sniffle. Well, we'll get to that in just a second, but why we have them. But what happens, why is IgA causing a hay fever? Well, IgE, it'll bind to, it'll bind to the allergen by its antigen binding site. The deal is the Fc region, it's specific for receptors on mast cells. And so when you get IgE binding to mast cells, it causes the mast cells to release histamine, and that's why many people are miserable right now if you have hay fever, because you got a lot of pollen and whatever, grass seed and whatever out there right now that's getting into your system, and you have IgE that's binding to it. Yeah, so the deal is this, if you don't have, like my wife right now is really getting hay fever bad, and I don't. What's the difference? It's a little bit genetic, in that if you check my blood, I would have stuff that's probably binding to allergen, but when antibodies binding to allergen in me, it's IgG. My wife, she's making her plasma cells that are binding to these allergens. She's switching the class to IgE for some reason. Yeah, and so it's just, we both have plasma cells that can bind to this allergen. But my plasma cells are telling it to make IgG, and hers are telling her plasma cells to make a different class of IgE, which is not good, because IgE binds to mast cells. Mast cells are lining your respiratory tract. Mast cells are, in your eyes and stuff like that. It's causing histamine to flood those areas, and that's why you're having hay fever right now.
So what does HCl do with histamine?
What is HCl?
Yeah, antihistamine.
So what it does is there's different antihistamines. Some actually target histamine. A lot of times what it does is it blocks the sites on the mast cells. Okay, so why do we have IgE? IgE is not the Fc portion not only interacts with mast cells, but it also interacts with eosinophils. Do you remember what eosinophils? What are eosinophils' main job? Parasites. And so eosinophils got to hook onto those parasites somehow. And so what happens is eosinophils, I don't have a good picture here, but so if this is a parasitic worm, for example, you have IgE will bind to it. And with eosinophils, let me label this. Eosinophils will have a receptor for the Fc portion of IgE. And once it binds to IgE, then it will degranulate, and you get this worm covered with eosinophils. That way, it'll kill the worm. Okay, so what's really cool about this, what we found out, is in areas where there's a high parasitic load, guess what? The population really doesn't have very many allergies. In populations where there's not a very good parasitic load, there's hardly any parasites around, or people don't have parasites, then there's high allergies. Kind of crazy, huh? And so it's like your body is making these IgE antibodies, and if they're not being taken up by eosinophils, then they find their way to the mast cells and cause allergies. Yeah, kind of interesting. And so for IgE, make sure that you know that it's involved in allergies and also in parasites. And yeah.
So do like anti-allergy medication have anything to do with
So if you take an anti-allergy medication, what it's going to shoot at is the histamine. It's all, it's going to be, it's going to be looking at the cytokines that are causing an inflammatory response like histamine. The best thing to do is to get allergy shots. The difference between allergy shots and the medications is allergy shots. So, for example, if they find out that you're allergic to cat hair or something, you have to be around cats, get an allergy shot, because what the allergy shot is just IgG antibodies to cat hair. Because then what will happen, if this is the allergen, you guys tell me, okay, why getting IgG antibodies, why would that stop the allergen? Right, and we call that, when you start covering this with antibodies like this, let's start covering that, and these are all IgGs, for example. If you cover something like that, what do we call that? Yeah, you neutralize it, right? So that's one of the 6 responses of antibodies. So by getting allergy shots, you neutralize your allergen with IgG. And that's what's, most people who don't have allergies, that's what they're doing naturally.
So how does that last?
6 months. But usually if it's a seasonal thing like hay fever, that gets you through the season.
So allergy shots won't help build immunity, just give.
Yeah, it just gives you passive immunity. Okay, the last one here is IgA. Most abundant, AB stands for what? Antibody in the body. Got a question about that?
So the other one was the most abundant in the blood?
Good, you're good. Yeah, most people are like, wow, but you got it. Yeah, exactly. So if you're looking in the blood, IgG is the most abundant in the blood. But if you're looking at the whole body, IgA. And you're like, well, where is IgA? It's not in the blood. IgA is on your mucous membranes. Your mucus is just chock full of IgA. Anything that's being secreted by the body has IgA antibodies. So, for example, breast milk, saliva, tears, mucus, all loaded with IgA. Yeah.
So you get a cut and it's oozing the IgA bacteria.
So that's oozing, that's the exudate coming in. So I'm thinking it's mostly IgA because that's coming from the blood. But anything that's coming from glands is going to be IgA. You know what? I'm going to tell you this, but I'm going to tell you that this next bullet right here, or the next 2 bullets, you don't need to know for the exam. Okay? But I just want to tell you because it's kind of cool. IgA has 2 different forms. It could be a dimer or a monomer. If it's found in the blood, it's only going to be one. It's going to be a monomer. If it's found in mucus or breast milk or something like that, it's a dimer. And it has to do with, anyway, it's not on the exam, you guys want to know more, take a immunology class. Or we might even talk about it in But just, so those two bullets right there, so you have 1 and 2. The third and fourth bullet is not on your exam. Okay? You're not held accountable to it. Plasma cells that release IgA antibodies are concentrated along the mucous membrane surface. Big thing that you want to remember for the exam, so I'll ask you this, is provides passive immunity to infants through mother's breast milk. So you get some IgG passive immunity through the placenta, right? And theoretically, those antibodies are going to last in the baby for about 6 months. But remember, as time goes on, those antibodies are going to diminish as they in number as they go. So by 4 or 5, especially by the end of the 6 months, they're not going to have very many antibodies left. So how does mom keep protecting the baby? Through breast milk. And so the breast milk is loaded with IgA, and also now mom and baby are usually close together all the time. So whenever baby's exposed to something, mom's exposed to it. So mom is making up this immune response, and baby doesn't. The baby doesn't actually gain a complete immune response until about 6 months. That's when the baby's immune response finally starts to kick in. And so that's why, if you see somebody moms who are careful where they take their babies out the first 6 months, probably rightly so, because the baby's immune response doesn't get up and get going until about 6 months.
So are babies that are formula fed more likely to get sick?
Yeah, makes sense, right? So babies that are formula fed are more likely to get sick because this doesn't have the mother's immune response.
Are they also more likely to get allergies?
Yeah, and they're more likely, and I don't know the reason for that, but I've heard that's true. And they're more likely to have allergies to foods too. Yeah.
So is that where colostrum comes in? Because I know with calves, the mother makes colostrum for the first 12 hours or so, and then it kind of goes away.
Yeah, and that's the same. That's true with humans as well. The very first breast milk is different than.
Yeah.
I was going to ask, so with vaccinations, do they vaccinate before 6 months? And if they do, then do you know why that would be the case?
Yeah, they do vaccinate. They give some shots before the first 6 months, and that's just because the babies are able to mount a small immune response to it, and what they're giving them is stuff that they are more likely to get in the hospital. For example, hepatitis and stuff like that.
And that would be the active immune response.
And that would be the active immune response. One thing, how I remember this little trick, you guys heard of IGA grocery stores?
Yeah.
So if you're group in an area where there's an IGA grocery store, what do you go to the grocery store to get? Milk. So that's how I remember. IGA is breast milk. If it works for you, if not, like, what the heck they're talking about? Find some other way to remember it. Okay, so that finishes up chapter 17. Any questions? We went through a lot of different stuff. We have some time if you want to go back over anything on your study guide so far. Yeah.
Real quick, what was it you said about food allergies?
Babies that don't breastfeed are more likely to have food allergies than ones that don't. And I'm not sure, I've never really looked into it, so I'm not sure what that means. Can I just tell you something that's kind of cool? If you guys don't have any other, this is kind of cool. So what happens is that when you eat stuff, your body has to somehow become tolerant to it. So when you're eating different stuff, although those proteins and stuff actually make good antigens for your immune system. The problem is you have to get those proteins into the blood to get into your cells. So there's always kind of a question of how come you didn't make an immune response against all of the foods and stuff that you eat? Turns out that, there's a mechanism that causes you to become immune tolerant